back from lunch. I'm Caiti Smukowski Heil, and I'm a postdoc in my Presention lab at the University of Washington. And today I'm going to tell you a tale of love, loss, and redemption in Hybridity, of which this is a picture caught in the act. So since this is the start of a session on hybridization, I thought I'd start with this quote by a plant biologist, Warren Wagner. We used to make fun of Edgar Anderson, another plant biologist, by saying that he was finding hybrids under every bush. Then we realized that even the bushes were hybrids. So I think this quote kind of sums up a current shift in thought from thinking that hybrids were once very rare to now realizing that they're quite common. And I think a lot of us usually think of negative traits in hybrids, like hybrid sterility and hybrid inviability. But some hybrids actually can tolerate stressful environments and adapt to new conditions better than their parentals. So here I have a couple examples. The iconic Helianthus sunflowers studied by the Reesberg lab, where the hybrids typically live in conditions that are very harsh and unfavorable to the parents. Darwin's finches studied by Grants, where there's evidence that hybridization allowed species to adapt to drought conditions. And of course, the famous beer and snow example. Actually, this is supposed to be lager brewing geese, which are hybrids. So the big question to me is how does this adaptation in hybrids actually work? And we can get some clues of this by looking at past hybridization events. For example, here I'm showing over here on the left we have tobacco, and on the right we have Saccharomyces pastorianus, which is the lager brewing yeast. And can everyone hear me if I talk like this? So when we look at these hybrids, we realize that there's a ton of different mutational events that can happen, and this can range from changes in gene content, gene expression, copy number changes, etc. And one of the most interesting types of mutations to me is this genomic shuffling, which I'll be referring to as loss of heterozygosity. And loss of heterozygosity, when I say that, I mean where one species allele is lost and the other is retained, and this can happen either in copy-neutral fashion or non-neutral fashion. And we actually, by looking at a variety of hybrids, these types of events actually are pretty common. So why? Is this just genomic shuffling in the process of stabilizing the hybrid genome? Is this the loss of nucleolus segments to settle a hybrid incompatibility, or is this actual selection of one species allele with the other? So to get a clue of this, we really need an experimental system. And the experimental system I've been using is the budding yeast Saccharomyces cerevisiae, and its close relatives in the sense of. And this is actually a really interesting space for studying hybrids. Hybrids are found in both natural and artificial environments. I already mentioned pastorianus, and there's many others found, especially in fermenting environments. And we can additionally create them in the lab, which is what we did. We created a hybrid between cerevisiae and Umbarum, which diverged 20 million years ago and are 20% divergent at the coding level. And just to give you some context of how divergent this really is, the amino acid identity is about the same between human and chicken. So this hybrid is like a human-chicken love child. So we can take this really divergent hybrid and its parent and evolve them in the lab. And the way we do this is using this continuous culture vessel called the chemostat. And the chemostat allows us to exert constant selective pressure for hundreds of generations. And the nutrient, or the conditions that we like to exert are nutrient limitations that may mimic natural conditions or conditions found in industrial environments, glucose, phosphate, and sulfate limitation. After allowing them to evolve for hundreds of generations, various mutations occur, and at the end we can isolate clones from different populations. And I've done this for 34 different clones of hybrid and parental background and whole genome sequencing. To summarize some of the mutations that we see, one of the really common events that happens in evolution is changes in copy number, and this can happen on the level of chromosome amplification, partial chromosome amplification, or these loss of heterozygosity events that I was previously describing. And the thing that I really want you to take away from this slide is that very few of the loss of heterozygosity events are actually shared between different conditions. And this suggests to me that this isn't the resolution of hybrid incompatibilities. If it was a hybrid incompatibility, you might expect that regardless of nutrient condition, you would shed the same portion of your genome. And instead, I favor an alternative hypothesis, which is that one species allele doesn't want to fit. And this is a case of adaptation due to standing variation. And those familiar with experimental evolution might appreciate this is a little different in traditional microbial evolution experiments that usually start with a homozygous ancestral strain and sex is absent, so there's no recombination. And any adaptation that happens is by default the result of de novo mutations. Because we have this hybrid that's very divergent, we're starting with unique haplotypes, and therefore adaptation can occur via both standing genetic variation and or de novo mutation. So to test this, I selected a repeated example of loss of heterozygosity. And here I'm showing a copy number plot for 6 different hybrid clones from phosphate limitation. On the y-axis is copy number, and on the x-axis is chromosome 13 position. And the Barham genome is denoted in blue, and the cerevisiae is in red. And very clearly you can see, particularly these 2 plots, this loss of heterozygosity event, which is actually accompanied by amplification in cerevisiae. And it's a little harder to see here, but there is a loss of heterozygosity event in a very small region, right, on this end of chromosome 13. And then we don't see any events on this tree here. So it's repeated in 3 out of 6 clones, and those are taken from 7 different populations, of which 4 have this loss of heterozygosity event. So what is in this region? What could be driving this loss of heterozygosity event? So if we look at just the smallest region of loss of heterozygosity, it's only a 25 kb stretch containing 10 genes. And right away, there's a gene that jumps out, SO84, which is a high affinity phosphate transporter. It senses phosphate and low phosphate conditions. So we're like, okay, this is a good candidate driver gene for this loss of heterozygosity event. And we can actually test this using the awesome power of these genetics. And the way we do this is with a simple allele replacement experiment. How this works is you take one species allele and simply add homology to it and pop it into the genome of other species. So in this example, you have cerevisiae background that's cerevisiae everywhere in the genome except at this one locus where you have the Barham protein. We can then do this for all of our different strain backgrounds and all different combinations. So we have cerevisiae, Barham, and a hybrid. And we can actually test the fitness of each of these strains using fitness competition assays. And the way this works is by using a GFP tag strain and competing these individually against a tester strain. And you simply look at whether the GFP or the non-GFP strain takes over the culture. And this gives you an estimate of relative fitness. So in this example, you can think of it as we have a GFP strain, the cerevisiae, 2 cerevisiae go to 4 alleles. You replace one of the alleles with the Barham. you can see that the fitness decreases. Similarly, if you replace both service alleles with uvarum, your fitness is also decreased. In uvarum, if you have two uvarum alleles and then you replace one of them with service, your fitness goes up a little bit. And again, if you replace two service alleles, your fitness increases. And this is consistent with the service allele being under selection in this environment. So finally, we can look at the hybrid. And indeed, we see the hybrid starts with one allele each species. If we have two alleles of uvarum, fitness is slightly decreased. And if we have two alleles of service, we have increased fitness. So overall, this is consistent with selection on standard variation at the 084 locus of service. So today I've told you about when two genomes become one. I've shown you love, that we can create interspecific hybrids in the lab and use them to study evolution of hybrid genomes in real time. I told you about loss, where hybrids can shed portions of their genome and loss of heterozygosity events. And finally, redemption, by selecting on standard genetic variation, the hybrid can actually become better fit to a new environment. And of course, this is only one case of loss of heterozygosity, and we see many other examples in the genome. But we could apply this method or similar methods, and we're actually working to use more high throughput methods to look at this, to come up with a more generalizable conclusion about loss of heterozygosity across the genome. And with that, my lab in some resources, pretty fast, so the question was how many uvarum alleles did they test? So there's just one allele of uvarum, but we actually have the cerevisiae was actually a heterozygous to start with, and we actually